here is a muscle, okay? So, muscles are covered with fascia. Fascia means bandage in Latin, okay? Cover something, right? It's around it, okay? The inner portion of the fascia is going to be fat and areolar connective tissue. So adipose tissue and areolar connective tissue, okay? It's going to insulate, it's going to protect, and it's going to hydrate the muscle, okay? The outer portion of the fascia is dense irregular connective tissue, okay? And then it goes right into the tendon. The tendon is dense regular connective tissue, okay? Then it's going to be attached to the periosteum of the bone, the periosteum of the bone, okay, which is dense irregular connective tissue. So you got dense irregular, dense regular, and dense irregular. Okay, so like for example, for next week's lab test, the way I'll ask the stuff about the types of connective tissue, I'll say, what type of connective tissue is fat, right? That's animals, okay? I'll ask them, what type of connective tissue is a ligament or a tendon? Dense regular. What's in a deeper portion of the dermis of skin? Dense irregular, right? It's pulling forces or in multiple directions. So it's very, very straightforward. Very, very straightforward. But you got to look at it. You got to study the stuff. Okay. So right here's the muscle. Okay. So if we pull this out here, it's called the fascicle. I'm going to go over this a bunch of times. Okay. So this right here is just this. We're trying to get our bearings. Okay. If I pull this out, that's what this is right here. This is just this is a blown up version of this. Okay. This is the muscle cell. So if you see a muscle cell, okay, or muscle fiber, the word is exactly the same thing. Okay. So we looked at epithelial tissue. It looked a lot different than connective tissue, right? They both look different, and the structures are different. And they have different functions. This is the next tissue we're going over, right? The 4 tissues. This is number 3. Okay, this is muscle tissue. Then we'll go over some nervous tissue next semester. Okay. So anytime you see a muscle cell or fiber, it's exactly the same thing. Okay, because the cells are long. Okay. Next semester, when you see nerve cell or fiber, it's the same thing. The cells are long. Now, like a nerve cell could be 3 feet long, like the nerves that make up your sciatic nerve running down your leg. Okay, so don't get confused. Okay, so now the plasma membrane of the muscle cell, right, separates the cell's external environment from the internal environment. That is going to be called a sarcolemma. Okay, a sarcolemma. So instead of calling it a plasma membrane is called a sarcolemma. So anytime you see sarco, you know it's going to be muscle. Okay? All right. Then you have the inside portion of the cell, right? The inside portion of the cell. The inside portion of the cell, just like other cells, right? The other cells, we call it the cytoplasm, right? In the muscle, we call it the sarcoplasm. Okay, called the sarcoplasm. That's going to be the cytosol and all the organelles. Muscles have different organelles, okay? All right? They do have, like, mitochondria, right? A lot of them, depending on the muscle cell. Mitochondria, and these are the nuclei here, also, okay? So, but they also have what's different. They have these sausages here, right? They're called myofibrils. These are the contractile elements of the muscle. It's going to shorten the muscle. The muscle shortens, right? It contracts, right? So they're going to pull on that joint, okay? So muscle cells have myofibrils in them. Muscle cells have myofibrils in them, okay? Right? Okay? And these little hairs here are called filaments. We'll go on that a little bit. Okay? So now, each individual, so this is important, okay? Each individual cell, each individual muscle cell is surrounded by protective connective tissue. It's surrounded by a protective connective tissue, and that's called an endomysium. That is called an endomysium. Okay? It's like a little saran wrap that goes around each individual muscle tissue. Okay? All right, it's called an endomysium. When we talk about nerves next semester, it's going to be the same thing, but it's going to be called an endoneurium. Okay? Neurium means nerve, mysium means muscle. Okay? So now, we take a bunch of these individually wrapped cells, and we're going to hold a bunch of them together, and we're going to wrap it again. We're going to wrap it with a perimysium. Okay? So it's going to hold a bunch of these cells together, that perimysium. That structure right now, that structure we call a fascicle. That structure we call a fascicle. So what's a fascicle? It's a bunch of individually wrapped muscle cells, okay, held together by a perimysium. Okay, and that's called a fascicle. Now we take all these fascicles and we're going to wrap those together. We're going to wrap those together, okay? And that's that's gonna be called a epimysium, right? Epi means above, okay, epi means above. So all these muscle fibers, they take turns when you're at rest, okay, or if you're sleeping, you're not using a muscle, they're gonna fire every now and again, okay? They're gonna take turns contracting. This is what keeps us toned. This is what keeps the muscle toned, okay? But if someone gets like a spinal cord injury, okay, and they get no nervous function to those muscles, what happens to the muscles? Okay, they atrophy, they waste away, they shrink and waste away, okay? So that gives us some muscle tone. And what does it, we'll learn this next semester, is it comes from the inside part of your brain, the substantia nigra portion of the brain. It's called dopamine, it's a neurotransmitter. Dopamine gives you these nice, evenly firing of muscles. You don't even realize it, okay? You don't even realize it, okay? When you're walking or you're moving your arms up and down, right? You got flexor muscles flexing. When they flex, the extensor muscles have to relax, and vice versa. You don't even think about that, do you? You don't even know what the muscles are called yet, okay? Because we haven't done it yet, okay? This is what dopamine does. Gives you that nice fluid contractions of your muscles. Now, there's a disease called Parkinson's disease, okay? It's Parkinson's syndrome, okay? These people don't release enough dopamine, okay? So instead of having a nice fluid movement in their joints, they get like a cogwheel rigidity. Instead of walking nice and smooth, they have a festering gait. You ask them questions a lot of times, it'll be like this. They hear you, okay? They understand what you're saying. They know how to answer you. They just can't do it. Okay? They just can't do it. Okay? So we'll get into that more later on. Okay? So again, right? A nerve fiber, a cell, exactly the same thing. All exactly the same thing. Okay. Then we have each individual each individual muscle cell is wrapped by a protective connective tissue coating. That's called an endomysium. A bunch of those are going to be held together by a perimysium. And then we call that big structure now a fascicle. All the fascicles are held together by an epimysium. We call that a muscle. And that whole thing's surrounded by fascia. Okay? Now let's look at these little myofibrils now. Okay? So if I took a myofibril and I cut it, right? I did a mid-sagittal cut. Okay, it's going to have thick and thin filaments. Okay, the thick filaments right now are called myosin, and the thin filaments are called actin. I'll show you a picture in a second. Okay, now these filaments now, okay, or this myofibril is going to be sectioned off. Okay, it's going to be sectioned off, and those sections are called sarcomeres. So it's from Z disc to Z disc. They look like little Z's. Okay. So sarcomeres are along the line, the whole myofibril, okay? When we want to do a contraction, the thick filaments look like golf clubs, okay? They look like golf clubs. The thin filaments look like little yellow golf balls, okay? And the golf clubs hit the golf balls and move the sarcomere inward, okay? So it shortens it. When all the sarcomeres shorten, the whole myofibril shortens. When all the myofibril shorten, the cell shortens. When all the cells shorten, the muscle shortens. That's contraction. Okay, that's contraction. Okay, and look at this. Muscle cells, right? Depending on what type of muscle it is, it's going to be multi-mitochondriated. Okay, I don't worry about that right now. Okay, so this here is my myofibril. Okay, so just pull it up a little bit. This is a sarcomere, and right here would be a sarcomere, right here. So the sarcomeres are along the line of the whole myofibril. Okay, I don't like this thing up so high because I can't draw on it, but we'll get around that. Okay, so if I blew up the sarcomere, this is what it would look like. Okay, this is the thick filament. We're going to see it's going to be called myosin. The thin filament is going to be called actin. These are contractile proteins. We call these contractile proteins. Okay, we have an end line. It's called the myomesin line. They don't even tell you that in that book. It's a little bit too advanced, but it's called the myomesin line. Okay, and then we have titin filaments. These are structural proteins, okay? So what happens here is when the golf clubs swivel up and grab the golf balls, okay, they go like this. The heads of the golf clubs pull everything inward towards this end line, okay? So this space here gets shortened. So when that shortens, right, and all the sarcomeres shorten and all the myofibrils shorten, the muscle will shorten, okay? Then when you relax, these tight filaments are like little springs, it springs back, okay? So now how we get that whole muscle to shorten, okay, or that cell to shorten, it's not even on this, but attached to the thin filament, that active filament, okay, there's a protein that attaches that thin filament to the cell plasma membrane, to that sarcolemma. It's called a dystrophin molecule. It's called a dystrophin molecule, okay? So, that's gonna come off, so let me go back. So that dystrophin molecule, okay, is gonna be attached, like this, right? It's gonna be attached, it's gonna attach the thin filament to that sarcolemma, okay, to the sarcolemma, okay? So when these shorten, it's gonna pull on the cell plasma membrane and shorten that whole cell, okay? Now some people, and they have a cure for this now, but some people have a condition or a disease where they don't make this dystrophin protein. What do you think that's called? It's called muscular dystrophy. Muscular dystrophy. You don't hear about it too much anymore because I found a cure for it. So it's a comedian called Jerry Lewis. He had for years and years. He knew someone that had this, and he had a telethon. So every Labor Day, it was the whole weekend, he'd stay up trying to raise money for this. And he did it. They found a cure. So when I was a little kid, a lot of kids with braces on and crutches because of this. Okay. And you tell when the infants would have it, because they walk on their toes, okay, because we'd be contracting them, okay, if you walk on their toes, okay, we don't hear it anymore. All right, okay, so right here, right, the thick filament is myosin, okay, so know that, and the thin filament is going to be active, okay, so the thick filament has these little heads. That's where the ATP is. These heads, see the little binding sites here? These heads will attach to the golf balls. See the little binding sites there? See these little guys? Okay? All right. Well, now, we have to have regulatory proteins, or we'd always be in contraction, right? So we have regulatory proteins. The regulatory proteins are going to be the troponin-tropomyosin complex. The troponin-tropomyosin complex. This here, okay, holds the binding sites. So now myosin can't touch it, okay? So what we got to do is we got to get myosin to touch it, and this is where the nerve comes in. This is where the nerve's going to come in. Okay. So now, here's the nerve, okay, going into my muscle. These are all my, this is the cell. These are all my myofibrils here. Okay. So if we're looking at a they put these in. They put no tray. All right. So let me just. So we're going to go over nerves next semester, but I want to just give you a little taste of what's going on here. So this here is going to be a multipolar neuron. Okay, so neurons are the nerves. Okay, so if I close my eyes and somebody gave me this marker, right? I can feel the marker. Okay, that sensation is going up my arm. It's going to synapse in my spinal cord, go to my brain. Okay, some out of sensory area is going to tell me, okay, you're holding something. Okay, the association area is going to be a marker. Okay, so that's sensory. Okay, now I want to write. I want to do something. That's motor. So that signal is going to go from my premotor area, my presensory gyrus in the brain, go down my spinal cord, down the nerves of my arm, and I can write. So that's motor. So I'm going to draw a motor neuron right now. Okay, so this is going to be a multipolar neuron, so it's motor. Okay. Okay, neuron's a nerve cell. Okay. This is going to be for motor function. Okay. So now, right here, these guys here, these are dendrites. Okay, whoops, if I spell it right. Okay. This thing right here, this whole thing here is the cell body. Okay, and we call that a soma. Okay, we call that a soma. Right? So now we got this long thing that comes down here. Okay. This here is collectively called an axon. Okay. And it's going to go out into, it's going to branch out into axon terminals. Okay. And then at the end of the axon terminal, we're going to have these little balls here. Okay. These are called synaptic end balls. Okay. The synaptic end balls contain neurotransmitter. Whoops. Okay. They're going to contain neurotransmitter. In this case, it's going to be acetylcholine. We'll go over that in a second. Okay, so now, motor neurons, okay, motor neurons, for the most part, they're going to have this protective coating. It's called a myelin sheath, okay? So what that myelin sheath does, okay, it's going to insulate that nerve. It's gonna insulate the nerve. So if you have a wire going into a socket, okay, if you took the big rubber thing off of it, you have 3 wires. One's a ground, one's a hot leg, one's a cold leg, okay? Okay, so one's coming in, one's coming out. If you took the protective coating off those, you got the 2 copper wires now. If you touch the copper wires, what would happen? It would short out, or the spark would short out, okay? That's what this is preventing. And since it's myelinated, okay, it's called saltatory conduction. We'll do that next semester. It goes very, very fast. It goes very, very fast. So the motor function tends to be very, very quick. If I touch the hot stove, right? If I'm touching a hot stove, okay, that signal, right, that heat signal is gonna actually come up to my spinal cord and go right back down again. Okay, there is a signal that's gonna go to my brain to let me know I just got burned. Okay, but it's gonna go to my spinal cord and go all the way back, so I pull away. Okay, it's called the reflex. Okay, it's called the reflex arc. Okay, so as I'm pulling away, I'm like, oh man, I just got burned. Okay, it's a defense mechanism because the longer my hands are on a hot stove, what's gonna happen? More tissue damage. So it just pulls away. Okay, so it's very, very fast. That's what this is. Okay, that is an autoimmune disease that your body attacks that myelin sheath. Everybody know what that is? The nerves tend to short circuit on you. It's called multiple sclerosis. Okay, all right? And what you get is like a flaccid paralysis, where you just paralysis, you can't move, or spastic paralysis, where it's all tensed up on you. Okay, all right? Okay, so now, anything that's affected by a nerve, anything affected by a nerve is called an effector, okay? Okay, so anything affected by a nerve is called an effector, okay? An effector can either be a muscle, skeletal muscle, smooth muscle, cardiac muscle, okay? It could be a gland, sebaceous gland, okay, endocrine gland, okay? Or it could be another nerve. You know, it could be another nerve. So in this case, our effector is going to be a skeletal muscle. Okay? Because we're talking about skeletal muscle. All right. Now right here, you notice that my synaptic end bulbs appeared here. They don't touch this, right? Because you got a synaptic gap here. Okay, you got a synaptic gap. Okay, so they don't touch. So I'm going to blow up here. I'm going to blow up my synaptic end bulb here. So that's what they're doing there, okay? And this here is going to be my effector, okay? That's my skeletal muscle. Okay. Now in my synaptic end bulb here, I got little vesicles, okay? These vesicles are going to have that neurotransmitter in it. Okay, the neurotransmitter. Okay? So this whole thing is to stimulate that effector, that muscle. Okay? This is the nerve, that's the myelin sheath. Okay? This is my axon. Okay? Some like muscles have axon collaterals, so you got multiple of these coming over. What? So you can stimulate that. Okay? And this here's my synaptic end bulb, my axon terminal, and my synaptic end bulb. So I'm going to blow this up a little bit more. Okay? These are my vesicles. These are my vesicles. So now what happens is I get an electrical impulse coming down here. Okay? I get my electrical impulse coming down. Okay? And what's in these vesicles is that neurotransmitter. But I got to open up the vesicles. So what happens is, right, we got to get a cation in here, okay? So we have calcium that's outside this cell. We have calcium outside the cell. And so when the electrical impulse comes down, it opens the gates, okay? And my calcium could go in there. What the calcium does is it opens up the vesicles, okay? So now all my neurotransmitter can go into my cell. Okay, so what we want to do is, I know it's a little much, but try to hang in there. Okay, if I was to get a gauge on how much electricity is in that muscle cell, okay, remember muscle cells don't do anything, it's at rest. Okay, So if a muscle cell's at rest, it's negative 70 millivolts. So that difference in voltage gives us that opportunity to bring stuff into and out of the cell. Okay, bring into and out of the cell. So we're gonna have diffusion's gonna, right, or facilitate diffusion's gonna bring stuff into and out of the cell, right? So it's gonna be right on a lab test. Okay, so if I were to measure a cell, right, a muscle cell that's resting, okay, it's gonna be negative 70 millivolts. A millivolt is 0.001 volts. Okay, so now it's resting, so we say it's polarized. So it's not gonna do any work, because it's resting, it's polarized, okay? But what I gotta do is, in order for it to do work, we gotta depolarize it, okay? Or make it not negative no more, make it not negative. So we gotta get it up to plus 30 millivolts, okay, in order for this to work. In order for this to work, okay? So now, if my cell's in a resting state, it's in a negative state, right? We gotta get it to a positive state. So how can we do that, right? Well, we could stick in that cell, okay, we could stick in the cell a cation. Remember what's a cation again? Maybe we did it, that's why I had in chapter 2. It's a positively charged ion, right? The cation, so think I like cats, right? Okay? The most abundant extracellular cation is gonna be sodium. It's gonna be sodium. It's gonna be sodium, okay? So, if I get sodium, if I put sodium in my muscle cell, I can depolarize it. It could do work, okay? But I can't do that, okay? Why? Because I got these gates in here. I got all these gates, okay? All these little channels are gated. So I have to open them up. I gotta have that be selectively permeable, right? I gotta open it up for sodium. And this is where the neurotransmitter comes in. This is where the neurotransmitter comes in. So the neurotransmitter, which is acetylcholine, right? They say a big H, little ACH. So that's like a key, okay, that's gonna open up a lock, okay? So when that acetylcholine comes in, the neurotransmitter is gonna open up these gates and sodium's gonna come in. When sodium comes in, it's gonna depolarize that cell. It's gonna depolarize that cell, okay? So I just went from electrical energy to chemical energy, and now that's gonna be in that cell. So you're gonna get electrical energy again. You're gonna get it again. So let me blow it up a little bit more, okay? Calcium, they don't say it here, but calcium comes in, opens up the vesicles, lets the acetylcholine into that synaptic gap, and then that acetylcholine acts like a key, opens up the gates so sodium can get in. So that electrical activity now is in the muscle cell. We call that an action potential, or electrical impulse. Okay, we call that action potential. Okay? So now we have electrically gated. So just real quick, we're gonna go over this next semester. Anything that's electrically gated, we call it voltage gated. Okay, voltage gated. Okay? If it's chemically gated, okay, okay, we're gonna call that Lightning gated, okay? All right. Okay, so, okay, wait, what's your name again? Luis. So Luis, right? Luis, actually, he's like a billionaire. He drives around in a limo all the time, right? It's kind of cool. So, right, so he goes to his house. He's got a big gate around the house, right? So the limo driver can open a gate. He could do it with a little electrical clicker. Thank you. We call it in Massachusetts a clicker. Okay, so they get a clicker and they open up the clicker, right? And so the gate will open. That's electrically gated, right? Electrically gated. Okay, so one day, right, the limo driver, he forgets the clicker, right? And so he goes to the gate. He could get out and with a key, open up the lock. That would be chemically gated, okay? One day he forgets both. What's your name again? Luis. So Luis goes out, right? He's got to get in the gate, right? He gets in and he kicks the gate and he breaks the lock. He breaks the lock. That would be mechanically gated. That would be like pressure sense, vibration sense. We're gonna go over this next semester, okay? But in the case of your body, right, you open up that plasma membrane, right? You mess up the plasma membrane, it breaks it open, but it goes back together again once that substance is in. So those are the two gates, okay? And then there's also mechanically gated, okay? All right. So now that electrical impulse, okay, is in the muscle cell now, okay? It's what it looks like. This is 1,650 times normal vision, okay? So right here, right, that's my synaptic end bulbs there. This is my muscles, it's striated. Okay, I know it's hard to see, especially in the back. I don't know which pictures are so small, but that's how they get it to me. Okay, so this is my axon terminal. It's my synaptic end bulb. The little red guys are the vesicles. Okay, so the electrical impulse comes in, opens up. You see the little speckles here? That's the neurotransmitter opening up the gates. Now sodium comes in. So now that electrical impulse goes around the cell. These are called T-tubules here, or transverse tubules. In between the transverse tubules, you have an endoplasmic reticulum, right? Just like we saw in the cells, right? In chapter 3. In the case of a muscle cell, it's called the sarcoplasmic reticulum. Okay? Sarcoplasmic reticulum in a muscle cell contains calcium. Okay, so remember, we want to make the muscle contract. Okay, but we can't because the golf clubs can't hit the golf balls. Why is that? Because of that tropomyosin complex. It's covering the binding sites. Calcium takes the tropomyosin complex off the binding sites. So now myosin in the golf clubs can hit actin in the golf balls, and now you can get the muscles to contract. Does that make sense? What I've been talking about for the last half hour happens like 0.001 seconds to do. Ooh, it did it. Ooh, it did it. Ooh, it did it. Let's do it. That's how fast it does it. Okay? All right? So I got to get the tropomyosin complex. See here? See the little blue and I guess brown? We got to get that off the golf ball. In order to do that, I got to have calcium. Okay? Calcium's coming from the sarcoplasmic reticulum. It's the endoplasmic reticulum of a muscle, right? Sarco again. Okay? And that's gonna, that calcium's gonna flood into that cytosol and remove the tropomyosin complex. So remember I said, right, when we did bones, right, the osteoclasts break down the bone to put calcium in the blood. What did I tell you? We need calcium for what? An electrical impulse, right? Right? It's got to open up those vesicles and to make the muscle contract. That's why you need calcium. Okay? This is where it's coming in. So, you see the big space between this yellow and that? See that big space? Right? The calcium's removing the tropomyosin complex. Okay? So now the golf clubs can hit the golf balls, and look at the space here. It's slid it towards that end line. See the little blue thing? That's the end line. Okay? So now, when that happens, we get what they call a power stroke, and it contracts. Okay, we contract. Once it contracts, right, we have an enzyme that floods into that synaptic gap. Okay, what did we use? The neurotransmitter was acetylcholine. Acetylcholine. The enzyme that reuses it is called acetylcholine esterase, right? Ends in ASC. Acetylcholine esterase cleans up all the acetylcholine. Now the gate's shut, okay, and we pump sodium out, okay? It kind of comes like this. It's a little, we're going to get into this more next semester. When the sodium leaves, okay, the sodium gate's shut, and first potassium leaves, okay? So you don't get, so you get like a little slope down like that. So we're going to hyper repolarize, okay, then you're repolarize. Okay? You just repolarize it. Okay? So we're going to put it back in a resting state. Once it's in a resting state, we have sodium potassium pump potassium back in and sodium back out. Okay, so we go back to that resting state. We'll do that next semester. Okay? All right? But now what happens is that we got what we call an esterase, right? Cholinesterase here, okay, that's going to pump all the calcium back into the sarcoplasmic reticulum. Okay? And then that's how you get a muscle to contract. It's a little bigger, right? So the electron impulse goes in between those transverse tubules, right? Those T-tubules. Calcium's released. It removes the tropomyosin complex. Okay, the golf clubs hit the golf balls, and it moves it towards that M line. That's how you get a contraction. Okay, I'm gonna blow this up a little more. How does the those golf clubs swivel up? Well, they have ATP. Okay, and the ATP we lose a phosphate group. If we lose the phosphate group, we're gonna get energy. Enough energy to pull up to the golf balls, right? The actin, and pull it towards that M line. Okay? When that happens to all the sarcomeres, the myofibrils shorten. When all the myofibrils in that cell shorten, the whole cell shortens. When all the cells shorten, the fascicle shortens. When all the fascicles shorten, the whole muscle shortens and contracts. Okay, it contracts for you. Okay? So it's a lot of, that's why chemistry is important. Okay? And physiology is important. Okay? Now how do we get the ATP? Okay? Well, we have it in our muscles, right? We get it from glucose. Okay, so two places we store glucose, right? We said the liver and skeletal muscle. This is why. Okay? Now, if we're exercising, we could borrow phosphate off another high energy bond, which is called creatine phosphate, okay? You need about 15 units a day, but you get it from the food you eat, okay? So if you're exercising, we could pull the phosphate off and put it in I did some diphosphate, and we get the other phosphate, which we could cleave off and get energy from that, okay? All right. Next semester, we will do metabolism, okay? So metabolism, we'll go over on glycolysis and beta oxidation, and how we get energy, okay? But the best way we want to get our energy is from glucose, right? That's why we want to make ATP. Okay, we can get it from the food we eat, okay? Right? We eat fruits, we eat vegetables, right? We eat wheat, any type of carbohydrate we eat, and we will break it down to simple glucose, okay? So if you're in a feeding state, right, you just ate, maybe a little while ago, that glucose is going to be in your blood, okay? Your brain triggers that, and say, okay, we got glucose in the blood, we got to bring it into the cell, okay? So your alpha cells will release insulin, insulin pulls the glucose into the cell, okay? We'll do this again next semester, okay? Now, if you haven't eaten in a while, all your excess glucose we store, okay? We store as glycogen. Where we store it? In muscle and the liver. So if you haven't eaten in a while, okay, we still got to fuel these cells. Okay, alpha cells will release glucagon. Glucagon will break down glycogen. Glycogen is like 100 glucose molecules long, okay? So break down the glycogen into glucose. Glucose will go into the cell, and in the cytosol of the cell, we will split it. Because we got to get it to go into the mitochondria of the cell. It's too big, okay? It is a 6-carbon sugar. It's too big, can't get in, okay? So, we will split it in a process called glycolysis. Glycolysis, lysis means to split. We're splitting the glucose into 2 pyruvic acids. If you see in a book, pyruvate, it's the same thing, okay? So now those 2 pyruvic acids could go into the mitochondria, okay, and then into the Krebs cycle and the electron transport chain to make electrons to make ATP. Okay, that's all we're doing with that. Okay, now, glycolysis, you do not need oxygen. Okay, Krebs cycle you do, electron transportation, you need oxygen. We're going to go over that in the next chapter. Okay, but glycolysis, you can get 4 ATP molecules from glycolysis. You can, okay, but you're only going to get a net gain of 2 because you need 2 to run the reaction. So you're making glucose, or you're making ATP, but to run this reaction, you're going to be using up glucose to make, using up ATP to make ATP, okay? All right, so any factory, right, that's going to produce something is going to have waste products, okay? So in making ATP, our waste product from glycolysis is going to be lactic acid. It's going to be lactic acid. So if you build up too much lactic acid in the muscle, what happens to the muscle? You're going to cramp up, right? That's why you do a cool down, okay? That cool down squeezes the lactic acid out of the muscle, puts it back into the blood, right? From the blood, it goes back to the liver, and the liver puts it back together and makes new glucose. It's called gluconeogenesis. Genesis means to make, neo means new, gluco means glucose. And we will do this next semester. It'll be a lot more understandable. Okay, you guys are looking at me like I'm out of my effing mind. That's why I'm like, nuts. But this is actually very important because the metabolic diseases like type 1 diabetes will cause this. Okay, this the mechanism isn't working, so we're going to jump over glycolysis and go right to beta oxidation, start breaking down fat too quickly. Okay, it's actually pretty cool. So when you get your biochemistries and stuff, you're going to go over this. Okay, again. All right, so now the preferred way, right, that we want to get energy is pyruvic acid from glycolysis, breaking down glucose. It's going to go into the mitochondria. Okay, say we haven't eaten in a while. Okay, or we're exercising a lot, or it's at night. Okay, we haven't eaten. Okay, we could break down fat with a process called beta oxidation. Okay, we can break down a fatty acid, like triglycerides and glycerol. Okay, that could go into the mitochondria, and we can get energy from that. Okay, again, the byproduct of glucose metabolism is lactic acid. The byproduct of fat metabolism is ketones. Okay, if too many ketones get in your blood, you can't filter them out through your kidneys. Okay, you're gonna go into an acidic state. Okay. Right? Amino acid is to break down a protein. We could get energy from that. That is totally not the preferred way to do it. Okay? This is if you haven't eaten in a long time. Okay? Or if you're starving yourself or somebody else is starving you. Okay? You start breaking down protein very quickly, you're going to get ammonia. Okay? That's going to be a byproduct, it's going to affect your brain. Okay? Now, once we go into the mitochondria. It's called the citric acid cycle or the Krebs cycle. Hans Krebs developed it, okay? So they named it after him, okay? We need oxygen for this to run, right? In our blood, right? And a red blood cell carries oxygen. It's the hemoglobin molecule. But in the muscle, we also have a myoglobin molecule. So you have two ways that you can get oxygen, because the muscles are constantly working, okay? Especially muscles of posture, your neck muscles, right? The joint is very, very small. Your skull's on your neck, right? But your head stays up all day. Doesn't fall over, doesn't get fatigued. How long can I hold my arms out like that before I get fatigued? Not that long. I'll drop them, okay? So you're using a lot of energy in your postural muscles, okay? So now we're making ATP, and the most abundant intracellular waste product is CO2. We exhale it, right? We get rid of it, okay? We're gonna get metabolic water, right? I showed you that when we broke down or neutralized acid, right? We get metabolic water, and you're gonna get heat. Because when you work out, you sweat. You want to dissipate that heat. But sometimes you turn red, so you want to dissipate that heat. Okay, we don't test you on any of this, just in the beginning stuff that we did. So the endonurium, the perinurium, and the epinurium we test you on, and the cells we test you on. Okay, but this here, if you took notes, that's great because eventually you're gonna need to know this. Okay, right. This is a motor unit. A motor unit is where a nerve hits an effector. Okay, so that's what we went on in the beginning, right? So the green is for sensory. So if something's touching my muscle or my muscle moves, it's called proprioception. So we will test this next semester. So if you hold your hands out like that, right? And I lifted your finger, with your eyes closed, you can tell, I said, what did I just do? You lifted my finger. If you have a spinal cord injury or a brain injury, you may not be able to know that. Okay, so that's why we test this. Okay, they're very bad. Okay, so that sensory information, right, goes down a peripheral nerve, right, either up your leg, up your arm, wherever. Okay, then it's going to synapse, okay, it's going to go synapse, right? That means it's going to jump from one nerve to another. Okay, so you're going to go in through that intervertebral foramen, right, the spinal nerve, and once you go through that intervertebral foramen, the spinal nerve splits. Okay, one's posterior, one's anterior. Posterior we call the dorsal root, anterior we call the ventral root. So right here it's called the dorsal root ganglion. A ganglion is where the synaptic end bulbs of one nerve hit the dendrites of another nerve. Okay, the nerves can affect nerves. So sensory goes in the back, goes up the spinal cord in the brain and gets interpreted. Someone just spit on me. There's a fly on me. I'm holding something. Okay? So now we have to react to that. Right? So we send the signal from the brain down the spinal cord. All motor information goes out the front. Okay? Ganglion's right here. Okay, it's called the anterior horn. Goes out the front into the muscle, and the muscle will react. Okay, the muscle will react. So we will test all this next semester. It's actually pretty cool. A lot of stuff we test seems pretty silly because we're all normal. Okay? We're all normal. But if you had a brain injury,